what's your reaction to winning a Nobel Prize?
I'm delighted to be sharing a Nobel Prize. I think the cryo-EM field has been doing great things in recent years, and the only unfortunate thing is they have to give it to 3 people. So there are quite a few others that have contributed to the growth of it, including other people in the lab. So, I think the feeling is that the 3 of us who've been awarded the prize are sort of acting as on behalf of the whole field. And I'm particularly keen or pleased to know that Jacques Dubochet and Joachim Frank are sharing the prize. Jacques, because he was the one who essentially kicked off the field. He's the real father of the field. He invented this method of making specimens that we now use. And then since then, there's been lots of instrumental improvements, computer improvements that Joachim Frank contributed to. And then people in the lab nowadays, like Georges Cherez, who's developed some new software that's made a really big impact recently. Nigel Unwin, that I worked with, I wouldn't be doing EM if it wasn't for him. So there are lots of people we have to remember, but nevertheless, it's delightful for the field. And a lot of them originally are from, these 3 people are from Europe, Switzerland, Germany, and Britain, although Frank has been in America now for quite a few years. So it is, and then there's been a lot of contributions also from people in Japan, Yoshi Fujiyoshi. So it is a kind of worldwide effort that's just now come to fruition. So it's obviously timely that they decided to give it this year.
Your work has been credited with pushing your field into a whole new era, new landscape. What do you hope will be the applications for?
Well, we are, the people who've done X-ray crystallography or NMR or cryo-EM, I think we all think of ourselves now as structural biologists. And apparently that term was coined by Don Casper about 1960, but now it's caught on because people go back and forward. And then the idea of structural biology is to find out the structure of everything that you need to know, everything that's important in biology. And we're getting towards that. There's a lot of structures known, and then the big thing about cryo-EM is you can use it for determining structures of all the other unknown structures at the moment that have been intractable by the other methods. And then once you've got the structures, the idea is you know about the mechanism, you can fit it into a bigger picture, but you may also be able to design either small molecules or other biological molecules that will activate or inhibit particular functions of those molecules, either in humans or infectious diseases and so on, so that you can block or stimulate the immune response or block cancer and so on. So I think it does lead into great value in medical research. And of course, we are supported by the Medical Research Council. They've been running for 105 years, since 1912. And they were set up originally for tuberculosis and rickets. They've solved all those years ago, and now they're on to other things. So we've been lucky here, since Max Perutz got the first award from the MRC in 1947, to be having very long-term support, and they encourage us to try new things, go in new directions, and it's been remarkably productive here at LMB, but also in other parts of the UK. I think they had the Magnetic Resonance Imaging Nobel Prize about 10 years ago, so that was at Nottingham. So MRC and the long-term funding has been very important. And then LMB, obviously, it's a big lab now with 400 or 500 scientists in it. But the culture is that of Max Perutz, who had his own work, but he found clever people to work on their own things and encourage them to take risks and do long-term research. So we've got the MRC and then the lab sort of culture. And then we now have 4 divisions in the lab. They all have their own philosophy and culture and so on. And obviously I'm in structural studies, and that was probably one of the strongest divisions at the very beginning with John Kendra and Max Perutz. And still it's a good division, and we're really strongly supported by the division. And then within the division, there are workshops that make things for us. So we're reliant on sort of the management of the lab, the workshops, and all the people who work together. So it's a good environment. And with MRC, and of course we're placed in Cambridge. At one stage, they were thinking of moving to London, but again, Perutz and Kendra said, no, Cambridge is a good place. There's lots of other people to collaborate with and talk to. So it stayed here, and I think it was a good decision. Would you like to know anything else?
I was giving other people a chance. Clearly, this is related to the number of Nobel Prizes that lab has won. What's that heritage?
Well, I think I always say to people that there's peer pressure that drives us on, Richard is the latest to succumb to this and attempt something really difficult, really long term. It is revolutionary, this sort of stuff, because it allows you to understand where drugs bind and what they do to proteins. It is the outcome of many, many years of consistent support, and you cannot tackle these difficult problems unless you have the confidence that your funding will not suddenly stop in 2 or 3 years' time, and that's been very important. It's also very interdisciplinary. Rich is a physicist, but we have mathematicians, we have biologists, very much part of the team, and here we've always had that tradition, and that's very important. I should say too that we've made considerable efforts to get this technology out there to the rest of the world and to industry, both locally and internationally. And that is also part of our remit, really, is to actually have that kind of influence on the world.
Yeah, we had, in terms of new equipment, we had the very first of these new, quite expensive electron microscopes in 2012, and we had the only one for about 2 years. And we did open it up to all the other UK groups, and eventually there weren't as many as we thought. So we had actually Spanish and UK groups using it for a couple of years. And then the second one came, which is now at the Diamond Light Source, and they now have 4 of them. And I was just told earlier today at a meeting on cryo-EM in Leicester that the company that makes these state-of-the-art microscopes, they've now got 20 orders. So they've delivered about a dozen. So within a year or so, there'll be 20 of them in the UK. So it's really spread quite rapidly during the last 5 years. And of course, the rest of the world's buying them as well. I think they're up to about 95, including the UK. But I think in this, as in earlier types of structural biology, Britain was always very strong in this. Other countries are strong in other things, but structural biology is very strong in Britain.
I'd like to just add that the Medical Research Council deserves a lot of credit for this, for realizing many years ago that molecular biology would one day be important for medicine. Of course, in the early days it wasn't really, but now we've seen the huge impact that it's starting to have, and this kind of confidence that scientists left to themselves will come up with things that are worthwhile. It's been really valuable to us, and I think that's not something that you can rely on a government or a company to predict what is possible. That's really something that people like Richard have the foresight to realize.
I think there hasn't been a really phenomenal impact yet from any of the individual structures from cryo-EM. There has been from X-ray crystallography, there's been in the lab from monoclonal antibodies. They've spawned multi-billion industries. But there are quite good indicators that it's going to be that. And so about a couple of weeks ago, there was a paper by Georges Cherez, Michel Godert, Tony Crowther, and so on, Anthony Fitzpatrick, determining the structure of one of these filamentous deposits that are in the brains of Alzheimer's sufferers who've died from that disease. And it's not exactly clear yet what the role of that filament is, but it's quite strongly involved in either the cause or so that's one example. And then there was another one again from Georges Cherez's work where they determined the structure of gamma secretase. That was about 2 or 3 years ago, and that's an enzyme that cleaves the proteins that make some of these deposits. So I think neurodegeneration alongside infectious diseases and then controlling bodily functions, there are lots of targets that emerge out of structural biology methods. So I think we're quite pleased that it's contributing, but it'll get better, I think, in the future.
And these are, of course, international collaborations. I point out that the gamma secretase drug target in Alzheimer's was a collaboration with China. Shores himself, of course, is from Holland. We aim to do science here, but we draw on the rest of the world very much to do it. And I think that's an important realization when things like this happen.
And also, in addition to the international dimension and the fact that people are contributing ideas and methods and equipment from all over, although we are talking today, there are now about 100 people in LMB using this cryo-EM methodology, and the equipment is maintained and developed and enhanced by a team of people, and we have 2 or 3 people: Xiaoxue Chen, Christophe Sava, and Giuseppe Canoni, who are running things and making it easier for people who are not experts and just want to use it. So there's a mixture of sort of dyed-in-the-wool cryo-EM experts that are working at it and developing methods, and then others who are using it as a technique to further their own goals, which are often biological or they have a target of that kind. So we're dependent on being part of an international community, part of the strong UK funding with the MRC, and then having a community that work together towards similar goals.
But for today, I could just say we're very happy. This has been a really another great day in the LMB's history, and I'm very pleased that Richard has got this recognition, well deserved.
It's over to you, Lee. Yeah, well, I'm just going to basically take the two of you outside in some light, and I just want some post pictures with the two of you equally. I'm going to take you downstairs to the board where it has the other 10.
Right. Are there only 10?
I hope there's space for your picture at the end.
I don't know. I think they have.
The bubble behind you, Richard, is that relative to the award?
Well, it's part of it. I was originally a structural biologist doing X-ray crystallography. And then in 1973, I heard a talk by Nigel Unwin, who's here still, and he and I are about the same age, actually. And he's the one who introduced me to doing electron microscopy, although I was keen only on diffraction at that stage. And this was the molecule that we worked on in 1973.
Oh, wow.
And although it took about 17 years to get it to this state. And then we stopped working on it about 20 years ago, actually. So this is a kind of historical thing now. But
that kind of brought you to the path where you are now.
Yeah, and then that methodology turns out, if you apply it to biological structures that are not crystalline, the images that you get from this new cryo-EM method turn out to be getting better all the time. And so now you can get the structure of virtually anything without having any crystals. And even though they're sort of intractable by other methods. So it is a very powerful method still being developed. And so we think all the remaining structures that we don't know yet in biology, there are now methods you can use. You can do crystallography or NMR or this cryo-EM method. So we think in a few years, I don't know, 5 years, we might know most of the structures certainly in the human biological population, and then in pathogenic bacteria and so on. So really it's quite an exciting time.
So had you not come to that talk here and sort of taken a different direction?
Someone else would have done it. Or, actually Nigel was thinking about how you can image proteins at that stage, but it was before cryo, and it was when you couldn't actually look at the atoms of the protein. You had to put negative stain and cover it with metal. But with time, the methods have been developed, and that's where Jacques Dubochet, the guy from Switzerland who was in Lausanne and who's now been retired for a year or two, he was recruited by John Kendrew, who was one of the founders of the LMB. Kendrew resigned his position as head of the division that I'm in at the moment and went to be the director at EMBL in 1973. And one of the areas he identified as being important was cryo-EM, and he persuaded Jacques Dubochet to be a young group leader at EMBL, and also specifically to work on cryo, liquid helium electron microscopy, and to study the properties of water when you freeze it. So in a way, John Kendrew, going back to probably 1947, when he started as a PhD student in the earlier MRC unit down in the old Cavendish, he had a lot of foresight and vision in many areas actually. And now EMBL is twice as big as the LMB actually. But they were, one of their earliest initiatives was this cryo-EM with Dubochet.
So the question has to be, where next?
You mean
where do you take the research from here, in terms of what you've been doing?
Well, I think our plan is to solve all the remaining problems, declare victory, and then retire.
But 5 years.
Yeah, well, 2 years actually. But there are other things, like there are quite a lot of people now doing cryotomography, and that is a little bit behind the kind of work that we're doing at the moment. But I think there's a lot of scope for further improvements there. But you know, the methods are going to get, but it'll become so routine probably. You know, Fred Sanger developed DNA sequencing, and it used to be done, you know, they would do 1 gel a day and a few bases and so on, and then people came and they were doing it all by hand, and then it was a big thing. You had to collaborate, you had to come here, you had to collaborate. But now it's been industrialized and so on. Now, nobody does DNA sequencing here. You send it off, and next morning by email, you get back the entire sequence of the whole plasmid. It's thousands of bases. So, you know, a suitable goal would be that, you know, you want to know the structure of something. You send it off tea time, and then next morning you get the structure back by email. That would be a very good success for cryo-EM. And then you know, we could do something else.
Richard,
Or the young ones could do something else.
we've got to wrap up, but we've had one question come in by email, which was, what were you doing today and what have you had to stop? What were you doing when you found out the news, and what did you have to stop doing for the rest of the day?
Well, either by good judgment or foresight or whatever, or by chance, one of the people who used to be a PhD student in the lab, who's now the head of structural biology in Leicester, had decided to have a meeting yesterday and today all about cryo-EM, because Leicester has just installed and inaugurated one of these new microscopes and has ordered another one. So they had a meeting to kind of raise the profile of it at Leicester. So I was there happily listening to talks by all our friends on cryo. It's quite a small, sort of fairly closely knit community. And then the phone rang at about 10:10. And I rarely get phone calls from Sweden, but I'm surrounded by the audience. So I rejected the phone call. And then it rang again, and I thought, well, I better. So I then went outside and called back, and then I chatted to people that we know. So I had coffee and chatted to them, and then I said, well, after 350 emails, I think I possibly ought to just call it a day. So then I drove back and had lunch with Hugh and so on, and then